All right, so it's 4 o'clock. So I'd just like to wait just maybe a minute more and see if we get any people join. There are a lot of people on leave and we're not in semester time. So it could be that people are planning to watch this as this is recorded. And I suppose, Ben, I should ask your permission at this stage if you're happy for us to record the seminar and share it with our audience. And you don't have to answer me now. You can tell me at the end if you prefer.
Fair enough.
But so, all right, well, I'll go ahead and get started anyway, and we'll go from there. So, yeah, you guys here know me. My name's Amy Diedrich, and I'm the Deputy Director of the Centre for Tropical Fisheries and Aquaculture, the CSTFA, and I'd like to welcome you to our seminar series. The CSTFA is based at the Babegu Yumba campus of James Cook University in Townsville, Australia, and we're a research provider for sustainable tropical fisheries and aquaculture research globally. Before I introduce our speaker, I'd like to acknowledge the Bindal and Wilgarukba peoples, who are the original peoples of this country from which we are hosting this meeting in the Townsville region, and pay my respects to their leaders and elders past, present, and emerging. I also acknowledge and extend my respect to the traditional custodians of the various lands on which all of you work today and the Aboriginal and Torres Strait Islander people participating in this meeting. So today it's my great pleasure to welcome Dr. Ben Mos. Ben is a marine science lecturer and Aboriginal man of Turrbal descent based at the National Marine Science Centre of Southern Cross University. Ben is funded by a Discovery Aboriginal and Torres Strait Islander Award and Discovery Indigenous grant from the ARC. And Ben's research looks at how we humans are altering waterways and oceans and impacting the organisms that live there by changing the climate, polluting our waterways, and catching too many fish with the aim of developing solutions to these problems. And Ben will be giving a presentation on alternatives to using toxic mercury in climate change research. So you're all muted for this presentation, but please feel free to put some questions in the chat box for Ben to address once we conclude. And you'll also have a chance to unmute and just ask your question in person if you want to do that once we conclude. So with that, I will pass the virtual floor to you, Ben, and thanks very much.
Thanks so much, Amy. Thanks for the invitation to speak to you all, and those of you who watch on video as well. I also want to acknowledge the traditional owners and custodianship of the land on which we meet, and pay my respects to the elders past, present, and emerging on the land of the Kumpengia people, and also want to acknowledge my ancestors and the Turrbal and my family, the Biripi people. Now, with these kinds of occasions, the guest is usually asked to talk a bit about what they do. Now, what I do depends on kind of who you ask. So as Amy so kindly put it at the start, so my research looks at how we humans are messing with the oceans and waterways and how we're impacting the organisms that live there. And kind of the things that I focus on are about climate change, about pollution, and catching too many fish, which of course I want to solve that, so I'm more focused on aquaculture. What I'm really interested in my research is trying to answer these big questions. So big questions like, how do we feed 10 billion plus people? So the United Nations predicts world population is set to reach 10.9 billion by 2100. The fact is we can't feed all 8 billion of us now properly. So where's all this extra food going to come from? My feeling is the solution has to be aquaculture. So growing more food in the oceans and waterways. And Professor Carlos Duarte and colleagues summarized the argument for aquaculture really nicely in their 2009 paper titled "Will the Oceans Help Feed Humanity?" And kind of their argument was twofold. Well, firstly, aquaculture isn't going to produce any more food than what it already does. And the fact is we're running out of arable land and fresh water is becoming more scarce. We can't rely on fisheries. It's not going to produce any more food. 33% of fisheries are already overfished. 93% are fished at or beyond their biological capacity. There's no more food coming from that. Their argument was instead we need a blue revolution, much as we saw the green revolution for agriculture. But the second part of the argument was, well, rather than farming at the top of the food chain in the oceans like we have been with salmon and tuna and things like that, we need to look further down the food chain. So farming down the trophic levels. And it makes sense, right? So only about 10% of the energy from one trophic level makes it to the next. It's totally inefficient to grow stuff at the top of the food chain. Instead, we've got to focus on the bottom. And that's kind of where my research kind of comes into that area. So one of the animals at the bottom of the food chain is sea urchins. This particular sea urchin is Trypnoustes gratilla. I've spent a considerable amount of my career working on this species, particularly with my colleague, Professor Simon D. Voyanen, both at Southern Cross Uni. And we've been developing the technology to grow these animals in captivity. So we've licensed this technology and we've successfully exported thousands of animals to Japan. Down here in the bottom right, you can see the orange gonads inside the sea urchin, and these are basically the only edible part of the sea urchin, and it's hugely popular in Japan, which is the main market, and it particularly goes into the sushi trade. But it's a high value product, which means we can grow it here in Australia as well. Now, one of the first things we looked at getting some of the basic information about how to grow these animals was looking at temperature. So we found that if you keep Trypnoustes too hot or too cold, they grow much slower than they would if you keep them at optimal temperature. They're kind of in this Goldilocks zone of 26 to 28 degrees Celsius is where they grow their quickest. And this is an urchin grown for the same amount of time at a slightly higher temperature, just a couple of degrees Celsius, and you can see it in fact didn't grow at all. So it's only that few degrees Celsius can make all the difference between having an urchin that is ready to harvest after only a few months versus an urchin that won't grow at all. And this also has implications when you start thinking about the effects of climate change for aquaculture as well. Some of the other basic information about growing Trypnoustes that was needed was like how many urchins could you grow in a tank or a set amount of space? How much water needs to be exchanged through their culture system? So this is the results of a 10-week experiment which tested the effects of density and exchange rate on growth and survival of juveniles. So the juveniles started off in the buckets like this, and only after 10 weeks they look like this. So you can see the massive growth rate of these guys. So these here are representative pictures of the Trypnoustes, in each are 3 densities and 3 seawater exchange rate treatments, and those were factorially crossed in all combinations. What we found was there was no difference in survival between the treatments, and survival was more or less 100%. However, there was like really strong effects of density and exchange rate on growth, spine length, consumption rates, food conversion efficiencies, all those things that you want to know about when you're trying to grow animals in aquaculture. And pretty much as you'd expect, so growth was fastest at the highest seawater exchange rate and slowed as the exchange rate was decreased, and growth was fastest at the lowest density and slowed as density was increased. All pretty pretty basic stuff, and you know, you see these patterns across all the characteristics that you measure, whether it be growth, spine length, consumption rates, food conversion efficiency, and so on. In some follow-on work that we did with adults, we found that density and exchange rates also have a limit gonad production as well. So urchins grown at high densities and low exchange rates had these much smaller and less colourful gonads than the urchins grown at low density and high exchange rates. And the effects were also apparent at the cellular level of the gonads. So in these two panels down here, the more stain there is, the better. Essentially, more stain, more nutritive phagocytes. So on the left-hand side here, where you've got that high density, low exchange rates, and much fewer nutritive phagocytes, and on the right, at a low density and high exchange rate, much more nutritive phagocytes. And these actually have an important role in the kind of the mouthfeel and the taste of the gonads. So essentially what this says is you've got to really control your density and your exchange rate. Otherwise, you're going to end up with gonads like this, which are not saleable at all. But if you get it right, you end up with some really bright, colorful gonads for which you'll get top dollar for. What was kind of unique about these experiments is that we took an innovative approach to working out what was actually responsible for driving these differences in growth and gonad production in different density and exchange rate treatments. So typically the approach would be to test the effect of individual factors by one by one. What we did was we used distance-based linear modeling to examine relationships between the performance of the urchins, so how quickly they grew, how much food they ate, all that sort of stuff. And then we also looked at, we also measured water quality parameters, and then we looked at the relationship between those two. What we found was that parameters like pH, total alkalinity, carbonate saturation state, and carbon dioxide levels were most correlated with growth and gonad production. So why was it that carbonate chemistry plays such an important role in determining the growth of these sea urchins? Well, sea urchins are a calcifying organism, and they use carbonate for the construction of their tests and their spines. And they take up carbonates, and during this process of taking up the carbonates to grow, it reduces the alkalinity of the water. And so when I say total alkalinity, I mean it's a measure of the ability of, in this case, seawater to buffer against acidification. So they uptake bicarbonate. The second thing they do as well is they also produce carbon dioxide. So they respire. The carbon dioxide is released into the water where it forms a weak acid, and then you get a reduction in pH as well. Now, importantly, because of the way the carbonate system works as this equilibrium reaction, more carbon dioxide in and the carbonates out, it actually acts to acidify the water and make it more difficult for the oceans to respire and to take up bicarbonates for growth. So you get this kind of double whammy effect. And we use the term biogenic acidification to describe the kind of overall collective effects of these sea urchins on the carbonate chemistry of the culture water. And this process may be generally overlooked. And in fact, the more we look for it, the more we see it in action in aquaculture systems. And we're actually seeing it in action. I just reviewed a paper recently that should be coming out shortly that looked at calcification from shellfish farms. They actually changing the carbonate chemistry of the seawater in their surrounding area and actually amplifying coastal acidification. So there's a whole bunch of implications about this work. But one of those implications is that it suggests that aquaculture of calcifying invertebrates such as sea urchins or mollusks will be highly vulnerable to ocean acidification. And ocean acidification being that reduction in seawater pH, which is associated with increased absorption of carbon dioxide out of the atmosphere. Now, I'm sure you're familiar with the keeling curve, which comes from the Scripps Institute of Oceanography. So interestingly, the curve hit 418 ppm parts per million carbon dioxide in the atmosphere just last month, which is higher than any CO2 level for 10,000 years. And predictions are carbon dioxide levels will reach levels that have not occurred on Earth for perhaps millions of years. So this kind of brings us to another big question, which is kind of related to what I was just talking about with carbonate chemistry, is, well, what are these effects of climate change in the oceans and for aquaculture going to be? And though we see carbon dioxide levels in the atmosphere rising, I would argue that it is in aquatic ecosystems where climate change is being felt the greatest. And we know that, for instance, more than 90% of the heat trapped by extra CO2 in the atmosphere has ended up in our oceans. And we've seen the pH of the ocean decrease due to the dissolution of carbon dioxide from the atmosphere into the oceans, forming that weak acid. Being able to test temperature and carbonate chemistry in aquaculture systems like I've just talked about has come in really handy for understanding the effects of climate change on the organisms that live in our oceans. And one of the animals that we work with in this context are the crown of thorns starfish. So for this particular study, we raised juvenile crown of thorns in the ocean conditions predicted by 2100. And then we measured how quickly they grew and how much coral they ate. Turns out the crown of thorns don't mind being in seawater with a lower pH. So these are animals that were in a pH 8.0, or more or less today's conditions, and a pH 7.6, which is kind of that level that's predicted for roughly 2100. So as you can see, the animals in the pH 7.6 grew much quicker. So, but what about the corals that the cots feed on? Well, we also looked at what happened to the corals that had been fed on by the crown of thorns versus corals that had not been fed on by the crown of thorns, and we kept those in ocean conditions for today and predicted by 2100. So it turns out the warmer temperatures that we expect to see by 2100 makes the corals more likely to die and slows their growth. And so that the coral takes much longer to be recovered after being attacked by the crown of thorns. And that's assuming that the crown of thorns just don't kill them outright when they're feeding on them. Yeah. All right. Another group of animals that I work on are freshwater shrimp, and I'm more interested in about the taxonomy, genetics, and ecology of these guys. What's really nice about working with these animals is I primarily do lab-based stuff, like I was talking about with the aquaculture and the climate change, but this stuff I get to go out into the field. And on one of these trips, I caught a crab, which I'd never seen before. So this guy down the bottom here, which was, turns out that species was Varuna literata, or the river swimming crab. And it turns out I found it in a place where it had never been found before, which is this further south location down here. So this was back in 2016. And the same species had only just recently been found by my colleagues just north of Coffs Harbour, so just here in 2013 and 2014. What was interesting was their previous range was restricted to the Brisbane River up here and north into the Indo-Pacific, if you look at the main map up here, so all through New Guinea, through the Indo-Pacific. So the crux of it is, though, that these crabs have moved about 300 k's further south than what they'd previously have been within the last few decades. And we know that they've been moving south because this area down here has been surveyed for freshwater crustaceans quite heavily, and they've never been found there before. So why are these crabs moving south? The most likely reason is climate change. So we know that here along the east coast of Australia, we've got the East Australia Current, and we know that that's getting stronger. The crabs release planktonic larvae that hang in the water column for weeks or months. And so it looks like it might be that the EAC is carrying their larvae further south. In fact, just 9 months before we published this particular paper, there was another paper published from South Africa that showed the same species, so the river swimming crab was also moving south there as well, more or less the same rate. Interestingly, they also have a south flowing ocean current that's getting stronger due to climate change as well. So kind of you've got this global pattern with this particular species. Is it a problem if these crabs move south? Well, we don't know yet, but we're concerned. And the reason is that the north coast of New South Wales here is a global hotspot for diversity of freshwater crayfish, like this guy here, particularly these spiny crayfish. And we know that we're particularly worried about the crabs bringing down new diseases or whether they might compete with these freshwater crayfish for food. So we need to keep an eye out for changes over the coming years. And we're particularly worried because quite a few of these species of freshwater crayfish are already under threat from introduced species, from habitat loss, from all sorts of different things. So that I don't think they need an extra stressor on top as well. I've kind of already talked about how the oceans are becoming more acidic and how they're heating up, but there's some other consequences of climate change that relate to the oceans as well. And one of those is as the oceans warm up, the polar ice caps are melting, and we're seeing a rise in sea levels. And we also tend to get more frequent and severe storm events. And this is something that we know too well here in Coffs Harbour. So this is actually a photo from a couple of years back where we had a really strong East Coast low. It brought some, I think it came in at a king tide as well. And so we had waves breaking over this, this seawall here, and it actually broke a whole bunch of infrastructure within the harbour. Consequently, the harbour wall has since been raised and all the infrastructure rebuilt. But it kind of gets to this point that with sea level rise and storms and things, we're going to have to upgrade our marine infrastructure in order to be able to cope with these changes. And since we're going to be doing it anyway, we wanted to kind of test out this idea that maybe we can use different materials in these upgrades to provide benefits for marine organisms that colonize these infrastructure, both now and into the future as well, as the oceans become more acidic and warmer. So what we did, we tested 3 materials commonly used in upgrades and in new marine infrastructure. And those were concrete, granite and greywacke. So granite and greywacke are two types of natural rock, and concrete being as we know, like a mix of materials. So what we did was we kept those materials in pH and temperature conditions predicted by 2100, and then we added larvae of Tritonius scrotilla, so the sea urchin, and then we followed how many survived and how quickly they grew under the warm and acidified conditions. What we found was that the urchins on concrete, sometimes, but not always, they tended to grow faster and had higher survival rates in the warm and acidified conditions compared to the urchins that were on the other materials, so on either granite or greywacke. So concrete seems to provide some benefits to newly settled sea urchins that help them cope with ocean acidification and warming. And we think the way that this happens is that the concrete leaches chemicals that buffer the pH around the surface, or that those chemicals are used by the urchins to grow their skeleton. And this helps them to ultimately grow faster and cope with those conditions. But we need to do more research to figure out exactly what is going on, so that maybe we can design concretes that do an even better job of helping marine organisms. And we also need to, of course, test the effects of the concrete in the field. But this is kind of a good news story. Like here is something that we can do that, since we're going to do it anyway, we can benefit both us and we can benefit the environment as well. Now, if you are working in the ocean climate change space, just like some of the experiments that I've just been talking about, you'll know that one of the most important parts of water chemistry that you need to measure is total alkalinity. So as I mentioned before, total alkalinity is a measure of the capacity of water to buffer against changes in its pH. So if we're talking about seawater, we're talking about the buffering capacity mostly comes from carbonates, but there are a whole suite of other chemicals that are involved as well. And these include things like boronates, phosphates, even nitrate will buffer against changes in pH to a small degree. So why do we need to measure alkalinity samples? Why do we need to measure alkalinity, and why do we take samples? The reason is, if you're doing these kinds of experiments, you need to know the seawater carbonate chemistry that you're working with. So you need to know pH, you need to know total alkalinity, and you need to know carbon dioxide levels. Now, if you measure two of those, you can work out the third one. And carbon dioxide levels in water are a total pain to measure, and just, if you actually want to do it, it's hugely expensive. So it's much easier and much more cost effective just to measure pH and total alkalinity, and then you can calculate carbon dioxide levels. That said, total alkalinity measurements are also time consuming to run, and it does take some time to set up and get it titrated, dialed in, so to actually run the samples and get the measurements. So it actually makes sense to collect your samples and store them until the samples can be run in batches. Now, in 2009, the Guide to Best Practices in Ocean CO2 Measurements came out, and that included standard operating procedures on how water samples for alkalinity should be collected and stored prior to analysis. Basically, the recommendation said that if you're going to do it, you collect the seawater and store it in high quality borosilicate glass bottles, and you poison it with mercury, and it should be right to sit there in the fridge for a while, and it should be okay. So that's what everybody, including our lab, did as well for many years. However, there's been some developments since then, and one of the things was that mercury was originally used to kill any bugs in the water, to stop them changing the water chemistry, and it originally came from people who were wanting to store water samples for measuring things like nitrate. But And that was back in the 80s. But since then, they've actually stopped using mercury to actually poison their samples. So we kind of thought, oh, okay, then why is it that we're still using it? We know that there's been some evidence in the literature as well that filtration doesn't affect alkalinity. There was some evidence that if you just put the samples in plastic containers, those are stable as well. And we'd actually done some pilot experiments indicating problems with stability when you actually use this standard operating procedure of glass bottles or vials and using mercuric chloride. There was also another question mark as well. So climate change in water doesn't only happen in the ocean, so that it happens in freshwater, it happens in lakes, it's happening in groundwater. And so people are starting to look at these other aquatic ecosystems to try and work out how climate change is affecting those as well. And then need to take total alkalinity measurements. So do these standard operating procedures for seawater actually make sense for all these other types of aquatic ecosystems as well? So this is what we actually set out to properly test. So here's what we did. So we got 4 sites which are marked by these red arrows, and these relate to 4 different types of water. So we've got some seawater, we've got some freshwater creek running down the back here, we've got a estuary here, or estuarine water, and then nearby that we had a site where we could collect some groundwater. And these were located around Coffs Harbour here on the east coast. And so we went to each of those sites and we collected 20 litres of the respective seawater type. Back in the lab, the first thing we did was to take 5 samples from the 20 litres, and we ran those samples to measure alkalinity. So the values that we got for those 5 samples, we considered that to be our benchmark control, with the idea being that if you're measuring alkalinity of any sample from any preservation or storage treatment, it should give you the alkalinity value if it's actually stable, if that treatment works. And if not, then the treatment's not actually keeping the alkalinity stable. That is, you should be doing something else. Then what we did, we looked at preservation treatments. So we followed the literature, the standard operating procedure using mercuric chloride. We did a filtration treatment, which was filtering just using syringe filters down to 0.45 micron. And then we had a filtration and a mercury treatment combined just to see if that worked better. And then we had a control treatment where we didn't do anything. We just put it straight into the storage vial. And then we stored the water samples in either glass vials or in plastic vials. And these glass vials had the same characteristics as the high quality borosilicate glass vials that are recommended by the standard operating procedures. And then we left those samples for either 0, 1, or 6 months. And all these treatments were crossed in all combinations. We had 5 replicates for each treatment. When you do the math, it's 120 samples for each of the 4 water samples, which is 600 samples in total. It takes about 5 minutes to run a sample. So if you run 600 samples, it's 3,000 minutes, and that's just running the samples. So you can appreciate kind of the enormity of this task, and probably one of the reasons why it hasn't been done until we've done it. So this is kind of the results that we got. The next couple of slides are going to look more or less the same, and the way to interpret this is look at the 0 here. So the 0 here represents everything's compared to our benchmark control. So if the treatment works, we expect that the change in alkalinity should be 0. So it should be more or less the same as the benchmark control. And any deviation from 0 is actually a change in alkalinity. And that indicates a problem. So this gray area indicates 2 standard deviations of the autogenic control, which is one way of measuring whether or not the sample's actually significantly different. The other way is these asterisks. These are the results of Dunnett's statistical tests, and what Dunnett's does is compare just the replicates in that treatment compared to the benchmark controls. So for instance, you shouldn't compare this one with this one. It only indicates this one compared to your benchmark control. So we've got our preservation treatments along the x-axis, and we've got our storage treatments, either polypropylene or the plastic or the glass vials in black. And then across the top, related to the panels, either initial, so time 0, 1 month of storage, or 6 months of storage. And in this case, we're looking at the seawater. All right, so what we found for seawater is that initially alkalinity is fairly stable regardless of how you treat the water. But after 1 and 6 months, you definitely start to see some changes, and they become quite apparent. Now, there were 2 broad trends that we saw. So in general, samples stored in borosilicate glass vials tended to have higher alkalinity than we might have expected. So here, for example, samples that were poisoned with mercury and stored in plastic tended to have lower alkalinity than expected. And so here we have all 4 water types. The other thing that was really apparent across all 4 water types, that the only treatment which was always stable, which always had alkalinity values that were the same as the benchmark control, was the combination of filtration and storage in plastic vials. And that's regardless of the time. So at 6 months, it was always the same, and across all 4 water types. And then we saw different changes in, as I talked about before, that there was this trend towards higher alkalinity if you stored samples were stored in glass vials, and lower alkalinity if they were poisoned with mercury. So the mercury concentration that we used was actually kind of at the upper end of the range that are typically used for poisoning alkalinity samples. So that kind of typically between 0.05 and 0.2% mercury within your sample. So what we did was another experiment where we again collected the 4 types of water samples and measured the benchmark controls. And then we tested the effects of a range of mercury concentrations above and below levels typically used. And then we measured the alkalinity after 0 and 1 month. And so you can see the 4 types of water here. So what we found was that for seawater and estuarine water, the alkalinity is fairly stable, except where you're using really high concentrations of mercury, kind of at that either the top end of the range that which people would normally use, or above that. For freshwater and groundwater, however, the effects were actually quite different and much more obvious. So even at concentrations at the low end of the range, you get these quite large effects in total alkalinity. And we don't know exactly how mercury reduces the alkalinity, but we suspect that it forms chemical complexes and actually precipitates out. And there's some evidence in the literature that this process happens more readily at low pH, which might explain why we saw more of a stronger effect of mercury for freshwater and groundwater than what we saw for estuarine water and seawater, given that these two have tended to have a much lower pH than these two. Finally, we wanted to see how the filter treatment would perform in a worst case scenario. So the reason why we actually preserve stuff is that that it's intended to stop biological activity that can produce acids or use up any of the chemicals that contribute to total alkalinity. And those biological processes need energy to operate. So our worst case scenario is where there's lots of chemical energy available in the form of dissolved organic carbon, or more specifically, glucose, at levels. And so what we did was we added the glucose at levels 10 to 15 times higher than the upper thresholds of any levels that we've actually measured in each of these water types. So even in this worst case scenario, filtration worked to preserve alkalinity, with a couple of exceptions. So for the groundwater here, total alkalinity fell by more than 1,000 micromole per kilogram, regardless of the treatment. And this was probably because this particular sample groundwater that we took had extremely high alkalinity, so more than 12,000 micromoles per kilogram. And if you compare that to seawater, which is 2,300 or thereabouts micromoles per kilogram, it gives you an idea of kind of how much alkalinity was actually in there. So what we think is happened for that, it was probably the some of the chemicals were actually precipitating out. All right. So if there's one slide that I want you to take from today, it's going to be this one. So in general, and there's a kind of a little asterisk there, if you're taking water samples for alkalinity measurements, all you need to do is filter those to 0.45 micron or less. You can use 0.2 micron if you'd like. Store those samples in plastic containers, and then store those containers at 4 degrees Celsius, i.e. in the fridge, and they'll remain stable for at least 6 months. Now, the little asterisk there, it's like there's always some fine print, isn't there? So the exceptions are if you're taking groundwater samples with very high alkalinity, so above about 4,500 micromole per kilogram, or water that has extremely high dissolved organic carbon. In those cases, filtration on its own probably not enough to keep the samples stable. All right, so why does any of this matter? What's the actual kind of reason that I want to talk to you about this today? And it kind of it centers around mercury. Now, mercury is great for putting in water samples because it kills everything, and that's the job it does, and it's highly toxic. So that's kind of why it's being used. But at the same time, mercury is arguably one of the most globally important pollutants. And so it's kind of hypocritical of us to highlight climate change in the oceans as an issue while simultaneously creating toxic waste when there's actually safer alternatives that are available. So that is just simply filtering the water. So it's actually, I think, a call for us to think about what impact that we're having as part of our research. So as a wrap up, I just got to say I'm really lucky that I get to work shoulder to shoulder with some of the best minds in my field. So I just wanted to acknowledge their contribution here, particularly Professor Simon Devoyanan, Professor Maria Byrne, and Professor Gio Turchini, as well as current and past funders. Yeah, so that's kind of what I do. I hope you found the talk useful and entertaining. Yeah, and if you want to get in contact, there's my email address and all that sort of stuff.
Great, thanks so much, Ben, for a really interesting and informative presentation. I'm going to open up the floor to questions, and I think we can all just turn on our videos and mics to ask questions since it's not a massive crowd. And I can see that Jan has got a hand up. So Jan, fire away.
Thanks, Amy. Thanks so much for your talk, Ben. That was really great. Yeah, I had a question about the freshwater crabs that you spoke about that are moving south. So Shannon actually, when she's not working for CSTFA, works for a project about rain-shifting marine species called REDMAP. So I was really interested to hear about those. And I wanted to know if you, so do the larvae of those freshwater crabs live in the ocean as well? Can they survive oceanic transport, or were you, or do you think that they're moving through freshwater bodies? It wasn't, I wasn't quite sure.
Yeah, so the larvae can survive in the ocean, and so this species is found throughout the Indo-Pacific and right across to South Africa, as I mentioned, and it's all in one species. So there seems to be quite a lot of mixing. So it seems like the larvae can travel quite far. So there's almost no way that they could be traveling via fresh water, given that we have all small creeks down this way, and they tend to flow straight into the ocean, and they don't tend to flood between them. That said, though, there was some big floods in Brisbane in the years prior to when we found these crabs. So it is possible that they were washed out of the Brisbane River and floated down on rafts, kind of with all the debris. And if you look at some of the satellite images from those floods, there were massive plumes of fresh water that actually came out of the Brisbane River during that time. So it's possible that they came down as either juveniles, probably little juveniles if they came that way, rather than adults, because the crabs that we caught weren't completely fully grown. So if they had been there for a couple of years, they would have had to start off fairly small. But yeah, so I think either of those two mechanisms. Kind of the point though is that they're making it down here and they're surviving, and the other thing is they're also making it through the winter. Which is the most important thing, because that means that they're here year round. They're becoming established. They're not just making it down during the summer and then being killed off.
Yeah, thank you.
I have a question, but I can't figure out how to do the hand up gesture. Sorry. Okay. Thanks for a great talk, Ben. Really enjoyed it. I hope I got this right, but yours, when you were talking about cots and the sort of relative effects of ocean acidification on the cots versus the corals, and I think I got it right that, well, I think my understanding is that you were saying that the cots were not so affected by ocean acidification. Is that right?
That's correct.
Yeah. So do you think that phenomenon applies to more broadly to echinoderms that are not so much? I mean, obviously coral is going to be massively affected by ocean acidification because it has a big skeleton, and you know, it's a very big part of the animal, I suppose, perhaps, you know, but echinoderms have skeletons too, and it's interesting that at least cots are not so much affected. And I now, drawing a bit of a long bow, I'm wondering, you know, how that might play into concerns about tropicalization of you know, temperate areas where you know, and some of the problems that we've seen sometimes in temperate systems where also there are problems with echinoderms there. And obviously algae, you know, is very differently affected from ocean acidification compared to corals. And, you know, there's this kind of concern that, you know, some of these southern systems will become tropicalized and maybe more like corals in the future. And I wonder if that maybe this sort of offsets that a bit.
So for the crown thorns specifically, one of the reasons why we think that they do so well is they're not as heavily calcified as some of the other echinoderms. If you think like a sea urchin has quite a large proportion of its body is made up of that calcified skeleton. Whereas for a crown of thorns, their spines, they've got some ossicles, but they're nowhere near as heavily calcified. The other thing that we found in that study is that those animals that were in the pH 7.6, they were larger, but they weren't as calcified. So it seems like they're having that negative effect of ocean acidification that's actually kind of hampering their calcification, but they can make up for it. What we think is happening is they can just, they can get enough protein to actually build muscles to hold their bodies together. And that way grow larger and more quickly. For other things like the sea urchins and that, there is some evidence that that the lower pH interrupts calcification and slows their growth down that way. But that said, lots of sea urchins are really slow growing anyway. And they're still having those kind of devastating effects on kelp as they move further south. We can think about like the Central Stephens moving down to Tasmania as an example of that. But because they grow so slowly, then they might just be able to cope with ocean acidification anyway, even if it is kind of limiting their growth a little bit. I think in general, though, if we're talking about the impacts of climate change, temperature is a big one for everything. And then, so I think that's the thing we've got to be worried about most. And again, that comes into corals as well, with bleaching. So I think that could kill off coral reefs far more quickly than ocean acidification.
Yeah. Thanks.
Have any more questions for Ben? All right. Well, in that case, I guess we're close to the end of our meeting anyway. And Ben, thank you so much for taking the time to give us this presentation. And we're really happy to have you. And I'll send you a follow-up email about the video and so forth. And yeah, otherwise, thank you so much. And thanks those of you who were able to be here in virtual person.
Yeah.
Thanks so much, Ben.
Thanks,
Thank you,
everyone.
Ben. It's a really great talk.
Yeah.